CAR T-cell therapy, or chimeric antigen receptor T-cell therapy, is an innovative form of immunotherapy that utilizes a patient's own immune system to combat cancer. The treatment begins with the extraction of T-cells, a type of white blood cell essential for immune responses. These T-cells are then genetically modified in a laboratory to express receptors that specifically target cancer cells. After modification, the CAR T-cells are multiplied and infused back into the patient's body. These engineered cells act like precision-guided missiles, seeking out and destroying cancer cells with high accuracy. This therapy has shown particularly promising results in treating certain blood cancers, such as acute lymphoblastic leukemia, ALL, and non-Hodgkin lymphoma. How CAR T-cell therapy works. 1. Collection of T-cells. T-cells are extracted from the patient's blood. These cells are crucial for the immune system's ability to fight infections and diseases. 2. Genetic modification. In the lab, T-cells are engineered to produce chimeric antigen receptors, CARs, on their surface. These receptors are designed to recognize and bind to specific proteins on cancer cells. 3. Expansion. The modified T-cells are multiplied to create a large number of CAR T-cells. 4. Infusion. The CAR T-cells are infused back into the patient's bloodstream, where they act like precision-guided missiles, targeting and destroying cancer cells. 5. Attack on cancer cells. The CAR T-cells recognize cancer cells by their specific proteins and initiate an immune response to kill them, potentially leading to rapid cancer cell destruction and, in some cases, complete remission. The pioneering work of Dr. Carl June. The development of CAR T-cell therapy can be largely attributed to the pioneering work of Dr. Carl June, a professor of immunotherapy at the Perelman School of Medicine at the University of Pennsylvania. Dr. June's research in the early 2000s laid the groundwork for this innovative treatment. His team's efforts culminated in the first successful use of CAR T-cell therapy in a pediatric patient, Emily Whitehead, in 2012. A real-life miracle, Emily Whitehead's story. Emily Whitehead's journey is a testament to the transformative power of CAR T-cell therapy. In 2010, at the age of 5, Emily was diagnosed with acute lymphoblastic leukemia, ALL. Despite undergoing multiple rounds of chemotherapy, her cancer kept returning. By 2012, her condition had worsened, and her doctors informed her parents that she had exhausted all conventional treatment options. Desperate for a solution, Emily's parents turned to the Children's Hospital of Philadelphia, CHOP, where Dr. June and his team were conducting a clinical trial for CAR T-cell therapy. Emily became the first pediatric patient to receive this experimental treatment. The process began with the collection of her T-cells, which were then genetically modified to target a protein called CD19, found on the surface of leukemia cells. The modified T-cells were infused back into Emily's body, and within days, her condition began to improve. However, she experienced severe side effects, including a life-threatening condition called cytokine release syndrome, CRS. The medical team at CHOP, led by Dr. Stefan Grupp, administered an experimental drug called tocilizumab to manage the CRS. Miraculously, Emily's condition stabilized, and her cancer went into complete remission. The impact of CAR T-cell therapy. Emily's successful treatment marked a significant milestone in the field of oncology. CAR T-cell therapy has since been approved by the FDA for certain types of leukemia and lymphoma, offering new hope to patients who have exhausted other treatment options. The therapy's success has spurred further research and development, with scientists exploring its potential for treating solid tumors and other forms of cancer.